All right, I think we're gonna get started here. Welcome everyone. My name is Jack. I'm the director of the Mariah Mitchell Aquarium. I am very excited to introduce James Elliot, who is the Green Crab Science Communication Director at greencrab.org. He got his undergrad at Salem State studying biology and holds a master's degree from University of Maine in forestry. He has spent time at the Cat Cove Marine Lab and Marine Biological Laboratory studying crustaceans and has published several papers on green crabs, especially population dynamics, and he's one of the local experts on the species. So we're really excited to have him here as part of Nantucket Green Crab Week. And without further ado, let's get started. How's it going, James?
Good, thank you, Jack. Should I share? Am I able to?
Yes, I'm going to give you host. All right.
All right, cross your fingers.
We should be all set.
All right, everybody see that? I think?
Yeah, we're all set.
Okay. Great, thank you to the Mariah Mitchell Association for allowing me to speak about this. This is allowing me to stay relevant with the green crab world as I've kind of moved off from academia and studying green crabs actively into industry. Right now I'm employed at New England Biolabs, and it's been quite an interesting year. And this talk has helped me provide a little bit of normalcy into my life, sort of revisiting a lot of the fun work that I did. Before I got started, I just wanted to say that I wouldn't have been able to do any of this if it wasn't for Dr. Alan Young at Salem State University. He got me into this and kind of guided me through all of this. And it's been a lot of fun working together with him and many other people that I'll mention at the end. But without anything else, I will get going. So what I'm going to talk about today is sort of an introduction to green crab life history. There'll be a brief review. As I understand it, there's a lot of different things that you're going to be, that you've already encountered this week and may encounter if you choose to stick with some of the future talks. But just as sort of a foundation for those who aren't familiar with green crabs, I wanted to provide some information on the invasion history and the ecological impacts that cause us to study these omnipresent crustaceans that seem to be just about everywhere. The thing that I hope to bring that is a little bit different is a lot of empirical data that is supported by a review that we were able to work on, that me and Alan Young, most recently, that looks at the life history and population dynamics of all of the populations of green crabs globally. So that'll be woven through, and I'll be presenting a lot of data that we generated over the last, I don't know, 7 years or so, 7 or 8 years. So that will include a trapping study that we did in Salem Sound in Massachusetts. There'll be a small section on habitat suitability analysis, and also briefly provide one slide talking about future work in greencrab.org, the organization that I'm currently involved with. So as an introduction to the European green crab, this, as the name states, this is a species that is native to Europe. This is basically going to be anywhere from the Northern Africa area around Morocco up through Norway and Iceland. So that's sort of its, what we can consider its native range. And it's pretty interesting what difference perspective makes. So about a decade ago, the biology and population dynamics of green crabs, especially chiefly in the United Kingdom, was reviewed in response to concerns that its abundance was declining due to overfishing, and there was questions about the sustainability of that fishery due to this overabundance in fishing. So there was talk of a maintenance program that might be needed to ensure that long-term sustainability. But just about everywhere else, there's a major issue with its invasiveness. It's considered to be one of the world's most invasive species. The IUCN, an authority on the matter, labeled it in the top 100. That really says something. In Massachusetts and New England and New Hampshire, in Maine, southern Maine anyway, they started arriving around the 1890s and 1900s, the early 1900s. They kind of moved their way up through southern Nova Scotia, Canada, and then reaching Prince Edward Island and Newfoundland all the way by 2007. So most recently in 2007, they've been sighted in Newfoundland. And that figure that you see on the slide that you there, that kind of shows the progression up in over the last 200 years or so. The primary invasive populations that are most heavily studied includes those on the Northwestern Atlantic, that includes us, New England, and also in the Northeast Pacific, where there is a heavily studied population of non-native species. So these separate invasions allow green crabs to be an excellent model for phylogenetic study. And that's how we get a lot of these dates as we start thinking, okay, how many invasions were there other than just documented accounts of there was this crab that looked like this 200 years ago. We're actually able to use some genetics in order to learn more about when these crabs came and where they came from at certain times. There's been a lot of talk on this, the ecological impacts. What we found in our review is that something that kind of stayed with me was that the green crab is considered to be an ecosystem engineer because it has the ability to modify the entire ecosystem in which it lives. This includes a variety of habitats, and we'll talk a little bit about habitat suitability. But these include mudflats, salt marshes, eelgrass beds, and also in some cases rocky shores where there isn't a competitor. In the Northwestern Atlantic, this animal has caused considerable damage to American oysters, scallops, blue mussels, and through competition, also rock crabs and lobsters. And even some plant life that's supporting an ecosystem for a very wide variety of animals, things like eelgrass. Most importantly, they've sort of, in our area up in Essex County, we hear a lot about the softshell clam and the effect that it has on the softshell clam. They have an absolutely devastating effect. And the reason we hear about this is because it's so important for the livelihood of a lot of individuals around Essex, Gloucester, Rockport, and this is kind of all throughout Massachusetts. But this is definitely the loudest concern for the green crab around our area. And this is because over the years, I mean, for the last 100 years, we've been noting that some of the poorest harvests of softshell clams have coincided with a high population of green crabs. And the inverse is also true, where we see very low levels of green crabs. We see really excellent levels of softshell clam harvests. And so this was mainly anecdotal, and it's been proven in the literature time and time again when we actually put some numbers to it. And the reasons for these fluctuations is mainly driven by the cold winters. So some of the concern is that as the winters stop getting colder, that there will be less dips of green crab populations that are going to allow for an excellent softshell clam harvest, thus supporting the livelihood of a lot of the generations that have built upon that in our area in Massachusetts. So one of the pictures here may seem a little random, and that's a duck. Sort of another area where green crabs can potentially disrupt the ecosystem is with eider ducks. There are parasitic worms that are pretty awful and can actually kill eider ducks and many other organisms. And there's these spiny-headed worms. That's what they're called. And the green crab, it actually doesn't affect them too terribly. but it will harm these ducks, which feed on crustaceans. So this green crab has also offered a nice vector for the spiny-headed worm to infect its hosts, chiefly eider ducks. So that's another way that may be potentially overlooked other than just eating things. As I mentioned earlier, this animal, because of its affinity for moving to different areas so quickly and so well, and really being established in a short period of time, as we've seen out west, is that there is a great opportunity to study population genetics within this species. This is one of the newer research frontiers. It's using genetic markers to really flesh out the population affinities and differences. So this is either the differences between two invading populations that may or may not have come from different sources. That will kind of be fleshed out and elucidated from looking at the genetics of these animals. And that could also mean taxonomic identification. So if you have two crabs that look alike, how related are they? Is one a problem, one's not? That you can use genetics to sort of tease those types of questions out. And just like I mentioned, the early goal of applying the study of genetics to learn more about green crabs was to determine the differences between closely related crab species. And I won't go too deep into this. When I originally developed this talk, there was more population genetics, but it didn't really seem to fit with the continuity of the more ecologically focused talk. But I will say that it's a very comprehensive overview in the paper that was in the sort of relevant readings in the biography. And if you are interested, there is a good amount of information there regarding population genetics. And this is such a well-studied animal in the field of population genetics over the last 20, 30 years or so, that it actually recapitulates the history of technological advancements within the field of genetics really well. So without talking too more about that, we'll get right into the fun stuff, the things that are a little bit more relatable for the majority of people. That was the trapping, and the goal with this was to investigate the population of green crabs in Salem Sound. And this was studied over a 3-year period from July 2013 all the way to June 2016, I think, using baited traps that were set out monthly at 5 different sites. And we measured a lot of different things. And on that slide, we really don't capture everything that we measured because there were some extra things that we measured that didn't even make it into any manuscript at the end. But things include seasonal catch per unit effort, the sex of each individual crab, the size, the ventral coloration, the role of the habitat variables and where these green crabs are going to be. This included abiotic factors like temperature, salinity, water depth, and the sediment size. And there's sort of a question of scale there, right? We were investigating things like habitat type, and then the interaction with 2 different ways that you can measure characteristics, and that's things at the individual crab scale. This is sex, color, carapace width, or characteristics of the population. For example, I see in this season, I see more crabs at this site. And so there's 3 different ways that you can look at this, and that's kind of what we were tackling. This, as you can see in that one picture that shows you sort of an aerial view of all the different sites that we looked at, these were all off of docks, and that was for the ease of, those for the convenience of being able to trap year-round. We didn't have to go out on a boat. The limitation with that is that typically we're trapping crabs within true estuaries where they're going to be a little bit more abundant. So this was just a gauge within this one river, and it was convenient because there was a lot of easy access to these. I froze. There we go. So one of the individual characteristics of crabs that we could measure, going back to the question of scale, was the sex ratios. And this was just to provide some sort of example for how we would take these individual characteristics and then compare them to global populations and make it relevant so that people would actually care about it. Because we're not doing this for fun necessarily. Well, we are, but we're doing this to actually inform people of something. The reason that sex ratios can be important and why we study that at all, whether there's more males or females in a given population, is because this is very important for the sustainability of a population. But we're not overly concerned with keeping the population of green crab high. You could ask the same question as how many males and females are there? And that certainly will contribute to the overall fitness score for that population, and would say, okay, this is a thriving population or this is a declining population. So that's one example of how you can translate sex ratios into a question that is actually usable. And as I say here, those sex ratios can vary greatly depending on the environment that you're studying. Males and females have very different habitat preferences. So where you're studying, if it's more conducive to a female habitat preference, you're going to have a female bias result. One example of that is that females are less tolerant of low salinity waters than males. This keeps a lot of the females in the subtidal zone where there's a consistent salinity and you're not having these waves and runoff of fresh water really changing things abruptly. Well, in comparison, males migrate in and out of the intertidal zone because they can handle it a little bit better. Let's see. I keep freezing here. Just to show you the variation that we see across the globe, what I did is I actually went through all of these published papers and calculated the sex ratios that were reported from all of their different trapping studies. And most of them do find sex ratios based on just trapping alone, similar to what we did in our own study. But what we came to a conclusion is that in all reality, there are just as many published papers that report a female biased male to female sex ratios as there are reported male dominated populations. They seem to be more common because of the way that people are trapping, or maybe the latitude that they're trapping in, and the geographical scale that you're considering a population makes a huge difference. And we believe that if you back that lens out enough, you would find this one-to-one relationship, especially with something that's doing so well and thriving so well. But we did see by compiling all of this data that there was a relationship between latitude, and that could be due to colder waters or warmer waters, skewing the sex ratio to the male or female end. But again, there may be other variables like seasonal and daily variations that could affect that as well, like how many people are trapping at night, for example. So that's something to consider. For our study, we knew that in Maine and New Hampshire, estuaries are both female biased. So then the question became, what about Salem Sound? Well, it's no different. From the 7,000 or so crabs that we trapped within Salem Sound as a result of our annual trapping study, we trapped much more as a result of combining other experiments that we did. But with that, it was about 75% females. And as I understand it, there was already a instruction on how to identify these critters, and that kind of recaps that. On the top there in that picture of the person holding these green crabs, on top is a female, on the bottom is a male with that narrower carapace telson. One of the most exciting things that we looked at a carapace width and many other things, but one of the more exciting things sort of stemmed from the ventral coloration. If this hasn't already been stated, green crab, the name green crab is a bit of a misnomer. If you flip them over, they can be any color under the rainbow, anywhere from green to yellow, to orange to red, and basically everything in between. So my advisor for my undergraduate research here at Salem State, I came up with this idea from reading Karen Lee's interpretation of how to do this in a low-cost, reliable way. How do you characterize the ventral coloration and quantify it really in a reproducible way? And so the brilliant idea of using Walmart paint swatches. So that's what Dr. Alan Young did, went to Walmart, picked out all the best looking paint swatches that may represent green crabs. And as you can see, it didn't do too bad. I mean, those colors are pretty close to objective as color gets. So we were able to look at all of these crabs that we would catch in each of our sites every month and then say, this was a 11, this was a 8, this was a 1. And by the end of 3 years, I could tell you which number it was just by looking at the crab. I probably still could. And one of those useless things that I picked up in college. So I had a lot of fun with that. And probably the biggest finding that we've had was associated with this color progression. And this is known that in the literature before we did any of this research, that as a green crab goes on and in between this intermolt period, because crabs molt just like all arthropods or these insects and spiders, all these things molt, right? So every time they're going to molt, their ventral surface is going to go through color transitions and all the way until the next molt. And the longer that they're in that intermolt period, the longer that's going to, the more red that crab will get. And this could be due to a few different reasons. It could be degradation of the ventral carapace as it's scraping along that rock, or if it's being just sort of wasted away in the salt water. It could be exposing this, the pigments that are underlying and the redder pigments. Or it could be just the opposite. It could be a thickening of the carapace that's, again, exposing different pigments or salts that are a little bit more red in their hue. So regardless of the reason why these things change color, they change color through their intermolt period. During our study, this is kind of a distribution of all the different colors that we collected through, and it was pretty uniform, especially for ecological data. Green, yellow, yellow was our pretty much our predominant color. But interestingly enough, when you look into the literature, the older literature, you see everything about green and red phase, but they were very clearly yellow crabs that really wouldn't fall into either of those categories. And we were sort of confused by that, but we carried on. In our, further on in our analysis, we looked at graphs like this one. And let me orient you if this is kind of a lot going on here. But on the y-axis, we have individual crabs that were in each subgroup. The subgroup represents the size class within the color, which is grouped together on the x-axis. So without staring at this for a while, I'll just give you the punchline, is that if you direct your energy, all your eyes to the orange and red groups, you'll notice that there is no crabs under the size of 30 millimeters. In fact, there's even a decreasing crab population between 30 and 45 millimeters as we get more red. And when we looked at a scatter plot, this was pretty obvious to us. And this is when we started to think with this, there's something to do with size and the color phase on the ventral carapace of these crabs. And on top of that, we recorded biofouling. And just as a way to reinforce the finding that as crabs age their carapace, they become more red. Well, this biofouling happens over time, and in the native and both the native and non-native populations, all of these populations become fouled with all these sort of epibionts, and that's what they're called. It includes like rock barnacles, slipper shells, bryozoans. Sometimes it includes tunicates, the sea squirts. And during this extended air molt period, the number of the epibionts increases as the crabs age without any sort of molting, as things just start to collect on it. So red crabs are by far the heaviest fowl, and that was certainly what we found from the data that we collected. So what's this magic number, 30 millimeters? There was this void in our data that said that any crab under 30 millimeters is not going to have any red pigment on its ventral carapace. It doesn't seem very significant, but we looked more into it because what else am I going to do? The literature from Maine, actually Barrell in 1982, discussed that 32 millimeters, or approximately 30 millimeters, is the cutoff size and carapace width for sexual maturity. And the conclusion that they came to was that the red phase is driven for a reproductive focus. This is indicative of a reproductive focus, whereas the green phase is not. This is more a crab that's interested in its growth, its physical growth, and not the growth of the population, like a crab with a reproductive focus would have. And if you look more into it, especially within the last few years, there's been a lot of noted differences between these two life stages or color phases. Things are different in aggression, things are different physiologically. Sometimes they have much different oxygen tolerances, salinity tolerances, and this was very interesting to me. And even more interesting, as I mentioned, was the neglect of the yellow phase. The majority of the crabs that we've found were very clearly yellow. And that, to me, seemed that it was a progression over to red. So are these differences clear within these different three phases? Could there be any information, more information gleaned from making a separate category for a yellow phase? So that was what we proposed in this 2017 paper that has been cited. And we sort of ran with that and spun off into another study investigating the endocrinology behind this. But let me take a step back and say, well, why the hell should we care what color green crabs are? Well, if I have to convince you, the green crab is a horribly invasive species. We've established that. For those who are interested in business, that means a high supply and really no demand. The culture of invasive species means we don't want them. To change that paradigm and sort of shift everybody's minds to that this is an opportunity, this was sort of the goal of the greencrab.org founding. So the idea is that you take a overabundance of green crabs and turn them into a culinary item. There's just one problem with that, is that when I sort of thought about this, and I said, well, maybe they just taste awful, and that's why nobody's had them. I cooked them over at the house when I was still living with my parents. I went to the trap, I took them out, I boiled them up like I would with lobster or crab or anything else. I tried them, and they tasted really delicious. The only obstacle was that I got about 20% of that crab edible. The dress out of the usable meat product for this animal is so minimal, I don't see why anybody would have any incentive to go out and buy it, unless you know exactly where to go or exactly how to clean that properly. So the obvious choice was to, well, this is a pertunid. This is a pertunidae. This is a swimming crab, just like the blue crab in Maryland. What do we do with the blue crab? We have a bioindicator that says this is when this blue crab is going to molt. We wait for it to molt, flash freeze it, and sell it for $5 a pound as a soft shell crab. That's a lot better than the proposed 20 cents a pound for a hard shell crab. And that's what we were looking at with subsidized trapping in Massachusetts. So this biological indicator, or this potential biological indicator of molting, could lead us to a model or a fishery that would create a demand where there is a high supply, thus managing the species effectively and also opening up a new area of the economy for Massachusetts, an area that is no foreigner to seafood economy. And the New Hampshire Sea Grant has done some really great work. I was sort of preparing for this talk and I stumbled across a lot of these guides, a lot of these keys that say, if you have a soft shell crab and you know where to find them, that's sort of kind of what we're doing. We're sort of, our goal at greencrab.org is to say, this is where to find them, this is what you do with them, this is how you cook them. So that way there are less obstacles in the way of shifting the culture towards understanding that the green crab can be used for more than fish bait. Also, something I thought was really cool is how do you prepare a hard shell crab? And then there's a bunch of other short information cards that are available through the New Hampshire Sea Grant. And those are collaborators with our organization, our nonprofit as well. So just to review a little bit of molting, I don't know if where everybody stands, sort of a crash course on molting. Every crustacean is going to molt. Every arthropod, insect, spider is going to have to molt during its life cycle. Specifically, green crabs are going to molt about 18 times. This is 4 during the growing phases. Well, it's a larvae. It's zoe and megalope stages. It's about 4 during those stages, and then another 14 after the larval stages. The molting cycle will consist of an intermolt period. This is termed diecdysis if it's a short intermolt period, or anecdysis if it's a longer intermolt period. It's not molting. This is a period in which it's feeding actively, followed by a shorter molting period when the old exoskeleton is shed to increase the body size. Different arthropods do it differently. Insects will take in air sometimes. Aquatic insects and crustaceans, they most often take up water to fill the void of the new shell while they digest the old shell and then crawl out and then take some time, around 2 to 3 hours for the entire process for them to come out of their shell. I see you have a picture right there of a green crab doing it. And every time they molt, they're going to increase their size anywhere from around 6-8 millimeters, which is pretty significant when they're an adult. And the mechanism of how they do it is actually very interesting. I won't spend too much time on it, but the key takeaways is that it's highly hormone regulated. Hormones are essential and absolutely necessary for all of these processes to be carried out. Ecdysone, appropriately named for the process of ecdysis, is the hormone that is going to be regulating a lot of this process. Unless they're juveniles, and then it's going to be another hormone called juvenile hormone in insects, a slightly modified, earlier, simpler version of juvenile hormone is called methylfarnesoate. That is the juvenile hormone for crustaceans, and the purpose of that is has many purposes, but it's going to be the driving force for molting in the beginning stages, in those first 4 stages in the larval phases of green crab. As the levels of methylfarnesoate are decreasing during those stages, decreasing levels show each are going to correspond with each stage of 1, 2, 3, and 4, fourth molt. So some other takeaways from this is that females have to molt to mate. Males are basically only going to molt when they're needing to grow or need to regenerate limbs, in other words, grow. In every case, this is going to be age and temperature dependent. The colder the temperature, the less likely it's going to molt. The older they are, less likely they're going to molt. And the process does peak at certain intervals in their life cycle. This is, again, all outlined in a recent publication that came out in January that I can direct anybody interested in it. As I mentioned, methylfarnesoate is the driving force of this. And there was reason to believe that this methylfarnesoate was going to be the regulating factor or the driving force behind the red pigment linking the inability to molt for some crabs. So what we did is we set up a study to test exactly that. What we did is we took about 120 crabs from November trapping, and we had a good selection of about, it was over 120. It was 120. I'm sorry. We took a good sample of male crabs, female crabs, and then a mixed population of crabs having them in 3 different tanks. And then we followed them over the course of 5 or so months, looking at them every 2 weeks, both for carapace width to see if they molted and how their color change progressed over that time. The reason that we separated the males and females is to test pheromone interference and observe the differences, because this could have been a confounding factor in anything that we were trying to observe. And there was an underlying goal of saying, well, how hard is it to culture green crabs if we wanted to set up a system like we have with blue crabs, where we're tracking them and eventually waiting for them to molt, flash freeze them, sell them. So in the middle of this study, once everybody was equalized, everybody being crabs, we took them out, took a hemolymph sample from each crab, the blood, and then analyzed it for the levels of methylfarnesoate. We compared male and female crabs within the groups of green, yellow, and red phase to see if there was a difference in the levels of juvenile hormone in each of these crabs. So it was predicted that there would be a higher concentration of methylfarnesoate in these redder crabs, because that was sort of released, that was published in 2008. But the testing hypothesis is, the hypothesis to be tested is that if yellow phase is indicative of an intermediate life stage, then the hormone levels should be different than the red phase. And that's pretty much what we found. The results did indicate that the color phase does share a relationship with methylfarnesoate levels. Furthermore, the finding reinforced the need for a distinction between the green and yellow phases. The ventral coloration also was shown to have a positive relationship with increasing endogenous methylfarnesoate levels and sexual maturity, because red phase is indicative of sexual maturity. This is something that's established. The mortality rate of that study does make the data questionable. So we believe that additional research is needed if either aspect is going to be useful for a bioindicator of molting or anything otherwise. So, water break. Next, I wanted to sort of shift to the importance of habitat suitability. Habitat suitability is very important because it tells you pretty much 2 things. It tells you where are the crabs right now, if I want to trap them, something we already sort of discussed, and then potentially even more importantly, where are the crabs right now is the reactive approach. You can also use this in a proactive approach, is where are the crabs going to be next? And this is going to lead to more proactive wildlife management in the areas of the potential invasion. Areas like Alaska that may see a great invasion over the next several years could prepare much better, and due to this work in habitat suitability, they have done just that. They have done a lot of great modeling that have shown this area would be a little bit more conducive to a green crab population, so we should be wary of it, be vigilant, and jump on it as soon as it comes. Some ideas to consider with habitat suitability is both abiotic and biotic factors. Abiotic would be things like the tolerance to temperature and salinity. Sort of touched upon that when we were comparing red and the male and female crabs. The availability of cryptic coloration against the substrate they're on. That's more important for the juvenile crabs. Availability of prey, predation, sediment size, depth. All of these things contribute. But what we know about habitat suitability already is that these green crabs are going to occupy a variety of sheltered habitats, such as estuaries, harbors, and potentially wave protected areas, but they're not able to survive in the open shore areas, the coastlines. For example, Winter Island at the bottom right of the map there had a really low catch because that in our study, and that was because of the exposure to very fast currents, waves. It was the site that was very open to the ocean. The currents were just not conducive, and this is mainly because these green crabs lack the ability to grip onto rocky substrates, unlike another invader in our area, the Asian shore crab. That crab has the anatomy and the gear to survive in those areas much better. So where they're going to sort of set up their house depends on the age. Different groups are going to prefer like juveniles are going to prefer more complex, smaller areas where they can hide out, such as mussel beds. There's going to be a concern with predators. They're a little bit more vulnerable. Younger crabs generally sort of after the juvenile stage are going to settle wherever they can. Something that is very important, or seemingly very important, is an area of complex cobblestone where they can be a little bit more camouflaged. As adult crabs move, they go into the lower intertidal zone and subtidal zones where they can shelter under rocks and boulders and even macroalgae and maybe eelgrass beds, and other fouling communities such as Spartina banks and estuaries. It's pretty wide range, as you can probably gather, of where they're going to live. But if there was any difference or slight difference or advantage that you could get in where you were looking for your crabs, then I would assume if you were trapping them for a profit, you would want to know that. So there's a lot of relevance to this type of work. For our study, we looked at the average catch per unit effort as a metric for how many crabs are in there. We weren't really able to extrapolate population density, because that is, you need a totally different experimental design for that, and I'm willing to take any questions related to that. But the catch per unit effort, which is every crab, so the reason for that is that every trap had a soaking time. The soaking time might have been a little variable, giving one trap at one site a slight advantage over the other. So what we would do is we would take basically catch per hour, or unit of effort, and take the raw number of crabs we caught, divide it by 48, and that way we get the catch per hour. Yeah, I think that's how we did it. I don't remember, it was a while ago. But it was a normalized metric, that's what's important. And looking at those data is that the fall always was the dominant year for each of the 3 years that we looked at these crabs. But there was a lot of high trapping variation within sites. And we saw that as an opportunity. What is special about 2 of the sites that make 3 of the sites sort of a lower abundance from regardless of the time of year or year that we were trapping? Well, if you look at the bunch of the sites, I kind of, spoiler alert, there wasn't a lot of difference between the salinity, the temperature, and other factors of exposure to the waves. I mentioned Winter Island had a slight exposure problem that we could attribute to low catches. But the 2 things that were drastically different seemed to be the sediment type and the depth at which we were trapping. Our Beverly Pier site, one of our most productive sites, was very deep. It was something like 20, 25 feet at high tide. Whereas our second most productive site, and some months our most productive site, sort of traded off, was our shallowest site. Sometimes I would find the trap out of the water when I went to it at low tide. So we'd have to move that. So there was high trapping variation, but we did have to do a directed study to say, okay, depth does not matter, so that way we could then tackle the sediment question. And guess what? No difference of depth. This was the combination of numbers from 2 different studies where we shuffled randomly the same trap with the same bait at different depths at one location and said, okay, that there is no difference. What we know about this already is that we know that green crabs can be found of depths up to 10 meters or more, but they're most commonly found at depths not really exceeding 6 or so meters, and their numbers diminish greatly after that. So in some locations, they have been noted to migrate into deeper waters during the winter. There seems to be a lot of inconsistencies in the way that this is reported, because there also seem populations that remain in the shallow subtidal throughout the year. And this is probably just depending on how much ice that area gets or the things that are influencing them, whether to stick around or not. Could be other things too. We did have a colleague, another professor at Salem State, that found that following a colder than normal winter, the density of the population was reduced, and many crabs were found dead in Spartina banks in an estuary close to here in Rowley. So that was kind of evidence that they did not migrate because they were dead. So these are sort of a this is sort of a table to show the variation of the different sites. And I wanted to call your attention to the column that displays the Wentworth size class. I put an orange box around the two sites to sort of to strain off your eyes, that were the most productive in catching crabs. Something that you can notice is they both had the Wentworth size class, or a standardized way of measuring the sediment size. This is the coarseness or the fineness of the sand. Is something really, really silty, or is it really more like a pebble? It may not matter to you, but it really matters to these crabs, seemingly. But I really can't go off of that just based on these data, because again, there were other variables that were affecting all of these different sites that, in my opinion, were not well controlled for as much as a directed study or a larger scale habitat suitability that would be looking at this in maybe across a larger geospatial area. So while I was at the University of Maine, I had the access to do such a geospatial analysis. And this is kind of the fun presentation, because I did all this without really leaving my desk. So the assumption here is that with caution, what green crabs consider to be a favorable habitat is assumed to be conserved across the coast of northeastern Massachusetts. To reinforce the results that we just saw from looking at the differences in sediment type across our different trappings, different sites in the trapping study, what I did is I took a lot of passive surveillance data. So anybody with the access to this type of software, or actually anybody now, can go into these databases, whether it's through the Division of Marine Fisheries or the Massachusetts Office of Coastal Zone Management, or USGS, or NOAA, or NASA, whatever it is. All of these files are publicly available. All you have to do is overlay them together and then test for differences yourself, because there's data available like you wouldn't believe out there. So that's exactly what I did. I went into the Division of Marine Fisheries website and downloaded a point file, which is exactly what it sounds like. It's a map that has a bunch of different points where green crabs have been spotted passively by either concerned citizens or biologists, and they were recorded. There was only about 150, 200 occurrences, but I figured this was enough for a small scale test that I was trying to do in a very short period of time. Combined with the green crab occurrences, I overlaid layers modeling distance to anthropogenic change. This, in other words, impervious surface like paved roads. The sediment type, which I found a file for, even in Salem Sound. The bathymetry, or in other words, depth. And also suitable prey habitat. Something that Division of Marine Fisheries needs to display is these files that show where's a habitat suitability for their prey. So what you can do is crabs need to eat. So if there's a high likelihood of soft-shell clams being there, for example, then it's probably a high likelihood that a crab would be there. So that was kind of the hypothesis. And that was because of the same reasons we wanted to sort of direct trapping efforts into a particular area. This was meant to be a model for the models in the future. How I went about doing that is with multi-band Landsat imagery and a supervised classification for anthropogenic change. What that means is that you just you go in and you say that a bunch of times and train the system to say this type of light pattern is what an impervious surface or paved road looks like, as opposed to an unsupervised change where they just assign different categories based on how they group things. This is how the system groups things. I also obtained that topobathymetry map to get the depth of water and interpolated all green crabs occurrence data from the Morris database, and then I analyzed these results. What this looks like in practice is something like this. This is way cooler than some of the other data that I've been showing. The multi-band Landsat imagery is shown on the left just to show you our study site, and I kind of made a high contrast image so you can see the exact outline of the estuary that we studied. Some attributes that I haven't mentioned is that there is this is a high salinity area. It's a pretty large coastal embayment and includes all of the towns listed above there, above the smaller map. The high salinity, even though this looks like a true estuary, where as you would go into the estuary, salinity would decrease substantially, that really isn't the case here. So with this in particular, it was because of the construction. So there was pretty uniform salinity all throughout, which was a bummer because we could show some really interesting data with that. Running a little short on time, so I wanted to just speed through this, but I'm willing to talk about any of these maps in more detail if interested. The short version of it is that when we looked at the 5 values, which are another way of quantifying the Wentworth class of sediment, they were averaged for each sediment type and then tested against the abundance of green crabs near them. This was a strong positive relationship. So there is something, in other words, to this fine sediment really attracting green crabs. It reinforces the study that we presented in 2017 that the reason that we had or part of the reason that we had a lot of high catches at 2 of our sites was really due to the sediment. And once again, depth showed no relationship, and the distance to land cover categories also showed no relationship with abundance. I just wanted to put in a quick plug for future work with green crabs. I'm going to continue to work as much as I can while I'm working at NEB. And I continue to encourage people to learn more about the invasive species and also encourage the use of green crab as a culinary item, as I'm still the acting scientific communicator, I think is what I'm called now. I'm not totally sure. For greencrab.org, we did have the organization, the nonprofit, did publish a cookbook. Mary Parks is going to be doing a cooking demonstration. She's definitely the expert, and it's going to be a lot of fun. I encourage everybody to look at it tomorrow night. And there is the link if you wanted to copy it down. And I also encourage you to visit our website. Thanks to everybody who made this possible. I want to thank my current employer, University of Maine, greencrab.org, and Salem State University especially. There's Dr. Young there. I couldn't have done this without him and some other people listed who were really helpful throughout the entire study. So thank you, and I'll take any questions.
Great, thanks so much, James. That was super interesting. I definitely learned a lot and already want to get back out there and look for more green crabs and see, how they're interacting with sediment and things like that. So that's awesome.
Awesome.
We did get a few questions. Let's see.
Can I see those, or where are they?
Yeah, so it's under the Q&A.
Oh.
On the bottom of your screen. Yeah.
Yeah.
So yeah, here's one. Having an automated way to produce green crab meat would be a great economic opportunity. Is there any work being done on a machine to remove the meat from green crabs?
Yeah, so when I was at the University of Maine, we had another person who was working with the greencrab.org. It was called something else at the time. And we tried putting it through a deboner machine. This is chicken deboner. It just removes that's kind of, it turns everything into mincemeat. And we thought that would be a good alternative and more economical and more efficient. And it looked horrific. I wouldn't recommend it, and was not it didn't seem like the quick solution that I thought it may be. Certainly the activity of, if we could just throw, pounds and pounds into a machine that would do all the cleaning for us, that would be a way better alternative and, way more efficient. But I know some people in Canada were looking into this sort of technology from like an engineering perspective, and I haven't heard anything since. And there have been a few other prospective ideas that I haven't seen go anywhere. But I'd love to hear and talk about any ideas relating to that.
Another question is, are there traps that will trap green crabs preferentially and to avoid trapping lobsters?
That's a good question. We did do a trapping study. We looked at what are the advantages of different traps, and one of my favorite traps was actually a modified minnow trap, where we cut out a hole so that it was a little bit larger, so that larger crabs could fit in. And for the space that it took up on a boat or small boat, if someone was going to be trapping these like recreationally and not like in a big, for a big business, I thought it was definitely the optimal trap for that purpose, because it was full every time we went through it. What we did is we just threw a piece of herring in there, and it caught everything. And it's about this big, comes apart, stacks really nicely. You can fit probably 100 on a 16-foot boat, no problem. And it's a really great way to catch green crabs. And legal lobsters aren't going to go in there, especially if you get a lot of green crabs in there already. There have been studies that show that lobster traps that are, what people will do. I think Wynn Watson did this at University of New Hampshire, where he put a GoPro inside of a green crab trap or a lobster trap, and he found that all these green crabs came in before the lobster, and then was starting to prevent lobster that were curious in the trap from entering at all. So the smaller the trap, I think the better, because you can fit more traps and you won't be catching respectably sized lobsters in that trap. But there's no way to exclude all of them that I know of. But I certainly don't know all the traps out there. So.
Great. Awesome. So another one, how do you age green crabs, and what is their sort of approximate lifespan?
We wrote this in the paper, and I don't remember. I'm looking for it. I don't know how you age them. I've never had to age a crab. I just know that the size is proportionate to the age in all cases. And it just gets a little hairy aging green crabs. Oh, right here. Lifespan is about 5 to 7 years. And that was reported in Oregon and Washington. And in the more northern waters, it was 4 to 6 years. So there's no data available for the other lifespans in other non-indigenous populations, but I'm not sure how they age them. Sorry, I couldn't be more helpful with that question.
And I don't know if you want to take a crack at this. I don't know. It might be a question for me, but it's from Alan Young. In our area, Asian shore crabs have outcompeted green crabs in rocky intertidal areas. Are Asian shore crabs in Nantucket waters at this point?
Well, I mean, the first part of that question, he knows the answer to. But the they have outcompeted in many areas. Asian shore crabs have outcompeted green crabs in many rocky intertidal areas. There was many areas in Ipswich, in Pavilion Beach in Ipswich. I've actually sort of noticed this over the years. When I first started going there, I could flip over every rock and there was a green crab everywhere. As a mussel bed came in and the area became a little bit more rocky as a result of the shift in the landscape, I noticed that it was completely dominated by Asian shore crabs. You can't find a green crab in that one location anymore. And this is the sort of the story with a lot of the rockier areas near us. As for Nantucket, I have no clue. I'm going to defer to you on that.
Yeah, so the Mariah Mitchell Association for about 15 years has been conducting eelgrass biodiversity surveys with seine nets on some of our field trips. And we find almost no Asian shore crabs. I think in our whole 15-year data set, we have maybe under 10 individuals collected in the eelgrass beds. However, they are extremely abundant in the man-made rocky structures on the island. So if you go snorkeling out by the jetties, you see tons of Asian shore crabs. There's this one little park behind the aquarium on the beach, and it's this grassy area that's built up on top of all of these rocks, and at a low tide you can flip over the rocks and there's like 3, 4, 5 Asian shore crabs under each rock. So they have conquered the very few rocky intertidal and rocky subtidal environments on Nantucket, but other than that, the whole island is sandy beaches, so they're not sort of abundant at large, but in those select areas, they're totally have conquered those places. So it's possible that those couple places may be source populations elsewhere in New England. I'm not exactly familiar with how long the larval stage is on that species, but it's interesting to find them in those select places.
Yeah, I mean, they definitely have a niche, it sounds like.
Yeah, at least here. I don't like the eelgrass. I don't know.
Yeah. Cool. All right. Was that all?
I think that's all of our questions. I wanted to thank you guys so much for attending, and thanks so much, James, for that really interesting talk. And stay tuned for the Mariah Mitchell Association and the Nantucket Land Council for the forthcoming green crab related events this week. And if you have any other questions about green crabs for myself, I'm happy to try to answer them or forward them to James as well. So I think we're going to end it now. Thank you so much again, everyone. Thanks so much, James, and have a great rest of your night, everyone.
Thank you.
Oh, James, I think you have to end the meeting because you're the host.
Where do I end it?
I think if you leave it, it will end it.
Oh, there we go. Okay, bye.